in this particular session, we will discuss some salient aspects about autosomal recessive inheritance. So the learning objectives for this particular session would be to understand the basic principles of autosomal recessive inheritance pattern. And I'll show you some pedigrees where autosomal recessive inheritance features are illustrated. And we'll also discuss some aspects about consanguinity and inbreeding coefficient. And one of the another aims of this particular session is to familiarize you with the concepts of compound heterozygosity, double heterozygosity, pseudodominance, and we'll discuss some aspects pertaining to genetic counseling in families with autosomal recessive diseases. So just to stress on some of the definitions before we begin discussing about autosomal recessive inheritance. So we all know that there are 46 chromosomes and there are 44 autosomes and 2 sex chromosomes. So a gene is actually situated at a particular locus on a chromosome. So locus is actually the location of a particular gene on a chromosome. So here on the left, you can see that there are 2 loci. Two loci, one is locus 1 and locus 2. So locus 1 has gene A and locus 2 has gene B. Now what is an allele? Allele is actually an alternate form of a gene which is present in homologous chromosomes. So here at locus 1, the 2 different alleles of gene A are capital A, this particular allele, and you have small a, which is this particular allele. Similarly, for gene B, which is at locus 2, the alleles are capital B and the small b. So when do we say that somebody is homozygous? When both the alleles in the homologous chromosomes are the same as in this particular scenario, we use the term homozygous. And similarly, when the alleles are different at a given loci, we use the term heterozygous. Now, dominant and recessive are actually terms which we use in relation to phenotypic traits. So phenotype is the physical characteristics, and we use the term dominant and recessive pertaining to phenotype. So when the phenotype is manifested in homozygous as well as in heterozygous state, that phenotype is said to have a dominant inheritance. And when the phenotype is manifesting only in homozygous or compound heterozygous state, then the phenotype is said to be a recessive trait. So even though we use the terms like a dominant gene or a dominant allele or a recessive allele, in strict terms, dominant and recessive are the terms that we should use to define a phenotype. Now coming to the term autosomal recessive inheritance. So the name itself is self-explanatory here. Autosomal means pertaining to the autosomes. And recessive means the phenotype will be expressed in an individual who is homozygous at a particular loci or who is compound heterozygous. So here you can see that here is a normal individual who has 2 wild copies at a particular loci, and the homozygous individual has 2 mutant copies, and the compound heterozygous individual also has 2 mutant copies, but the 2 mutant copies are actually different, and a heterozygous individual has 1 wild copy and has 1 mutant copy. And heterozygous individual in case of an autosomal recessive disorder is termed as a carrier. Generally, carriers are not will not have any particular phenotype. So this is a pedigree that apparently depicts a possible autosomal recessive inheritance. So here you can see that there are multiple individuals who are affected. So the affected individuals are actually shaded in red. And you can see that in the 5th generation, in the 4th generation, here there are multiple individuals who are affected. And here in the 3rd generation, there is actually consanguinity, which is depicted by these double lines. Now, some of the characteristic features that will help us in identifying autosomal recessive inheritance are, so as I said earlier, there will be multiple individuals in a particular generation who are actually affected. So this is in contrast with autosomal dominant inheritance, where multiple individuals and multiple generations will be affected. And in autosomal recessive inheritance, initially this was known as horizontal transmission, but that term is now not used anymore. And usually we can see here that the parents are carriers for this recessive disease and recessive condition, and they are usually asymptomatic or unaffected. And consanguinity may be seen, but it is not a prerequisite for autosomal recessive illness. And since it is an autosomal condition, or because sex chromosomes are not involved here, both males as well as females will be affected. What is consanguinity? So consanguinity is defined as union between two individuals who are as close or closer than second cousins. And these individuals inherit an allele from a common ancestor. And in case of an autosomal recessive disease, consanguinity increases the chance that both the partners are apparently carriers for a particular disease. And consanguinity is thought to precipitate very rare disorders. So even though we associate consanguinity with autosomal recessive disease, consanguinity is not the most common reason for common autosomal recessive disease. For example, in case of beta thalassemia, the carrier frequency in our population for beta thalassemia is actually high. So consanguinity is not the reason for occurrence of beta thalassemia. It is because of higher rate of carriers in the general population. So consanguineous union can be of different degrees. So we usually do not come across first degree consanguinity, and because that is otherwise termed as incest relationship. And then you have second degree consanguinity, where you can have a relationship between half siblings or uncle, niece, or aunt, nephew, and double first cousins. Then you have third degree consanguinity, where first cousins have a relationship. And so here, this is one of the common scenarios that we apparently come across. And you have fourth degree consanguinity, where first cousins once removed or half first cousins have a relationship. And you have fifth degree consanguinity, where second cousins have a relationship. So this is just to emphasize the fact that as the relationship is closer, then the chance that they share share the allele from a common ancestor also increases. Now, inbreeding is another term that sometimes is used interchangeably with consanguinity, but inbreeding in strict terms means departure from non-random mating. So here what happens is the individuals tend to mate with those who are genetically similar to them. And these individuals, if we ask them, they will deny that they are related, but they may be from a similar background or geographical origin. So inbreeding is another reason for occurrence of very rare autosomal recessive diseases. Now, we have to familiarize ourselves with another term known as inbreeding coefficient. So the probability that a homozygote individual has received both alleles at a locus from the same ancestor is known as inbreeding coefficient. It is the proportion of loci at which the offspring is homozygous and the offspring derives the alleles from the same ancestor. It is denoted by the term F. And we consider that consanguineous relationship or consanguinity is significant if the inbreeding coefficient is equal or more than 0.0156. So this is just to show the inbreeding coefficient in different situations. So here you can see that when it is a union between a double first cousins, then the inbreeding coefficient is quite high, which is 0.125. As in the case of uncle-niece relationship, where inbreeding coefficient is high, 0.125. In first cousins, the inbreeding coefficient is only 0.0625. So as the inbreeding coefficient increases, it just denotes that the proportion of the shared alleles is increasing. So this is another figure just to show the inbreeding coefficient in other different scenarios, like first cousins once removed, second cousins, and double second cousins. So as I said, so we consider a relationship as significant or consanguinity as significant if the inbreeding coefficient is actually more than 0.0156, equal or more than 0.0156. And as I said earlier, even in the definition of consanguinity, any relationship which is closer than, which is not beyond second cousins is considered to be consanguineous relationship. Now here is a family where there is a child, this particular couple, you can see that they are non-consanguineous and they have two children. So the first child is unaffected, the second child has beta thalassemia major. So we all know that beta thalassemia major is one of the common autosomal recessive conditions, which is caused due to homozygous or compound heterozygous variants in HBB gene. So here we need to understand something more about the term allelic heterogeneity. So not only one particular type of variation in the HBB gene causes thalassemia, there are different types of variations in the same gene that can cause the same clinical phenotype of beta thalassemia. So such a situation is known as allelic heterogeneity. So allelic means different types of alleles will be there, which will induce the same sort of phenotype. So here, this particular child was had compound heterozygous variants in HBB gene, where one variant was C.92 plus 5G2C, and the other variant was the 619 base pair deletion. So here we can see that the father and the mother each have the wild type allele, which is green, and they have one mutant allele, which is red, and the other one is blue. So they have two different types of mutant allele, but the child has inherited both these mutant alleles from one of the parents. So the child is actually compound heterozygous, and this particular situation where multiple mutations or multiple variants in the same gene can lead to a same phenotype is known as allelic heterogeneity. Now, some of the autosomal recessive conditions exhibit something known as locus heterogeneity. So here, one of the common examples for such a scenario is deafness, where variations in multiple genes can cause deafness, like multiple genes which are present in multiple different loci are if there is a variation, if it can lead to deafness. So here you can see that in this particular family there are two individuals, 2-1 and 2-2, who have deafness, and the individual 2-1 has deafness because of a homozygous variant in MYO7A, which is a particular gene which is responsible for causing deafness, and individual 2-2 has homozygous or biallelic variant in MYO3A, and here, these two, MYO7A and MYO3A, are two different genes which are present in two different loci. So, but none of their children are deaf because all of them are double heterozygotes. So they have inherited the MYO7A variant from father and MYO3A variant from the mother in heterozygous state. So this is known as double heterozygosity, and they do not have any clinical phenotype. Now, coming to the recurrence risk. So, in case of autosomal recessive disorders, three different scenarios can apparently happen. So, there may be a situation where two individuals are carriers for an autosomal recessive condition. So, this is quite a common clinical scenario. So, here you can see that these individuals are carriers for an autosomal recessive condition, and here only 1/4 chance one out of four chance is there for their offspring to have both the mutant alleles from the parents and thus being affected. So the chance of them having an affected offspring would be 25%, and 50% of their offspring would be carriers, and 25% of their offspring would have wild type allele. So apparently here 75% chance is that the offsprings are unaffected, 25% chance to have an affected offspring. Now, another common situation could be where an affected individual, like a person who has an autosomal recessive condition, actually has a relationship with a carrier individual. So here what happens is 50% of their offsprings would have both the alleles, would inherit both the alleles and thus become affected, and 50% of them would be carriers. So here on the right side, you can see that this is a family where an individual who has deafness due to deafness autosomal recessive 1A type has married a person who is also a carrier, and the carrier is asymptomatic, but two of their children are apparently affected. So here the pedigree may sometimes actually resemble that of an autosomal dominant condition, because it looks like the mother has transmitted the variant to both her children in a vertical fashion. So that is why this situation, or this pedigree is known as a pseudo-dominant pedigree. So here an affected individual, when an affected individual has offsprings with a unaffected carrier individual, the pedigree may resemble an autosomal dominant condition. This is termed as pseudo-dominance. Now, there is another scenario where both the partners could be apparently having an autosomal recessive condition. So, in such cases, all of their offsprings, or 100% of their offsprings, would be affected. Now, some of the common examples for autosomal recessive inheritance, conditions which follow autosomal recessive inheritance would be spinal muscular atrophy, beta thalassemia, and majority of the inherited metabolic disorders follow autosomal recessive inheritance pattern. So here is a family where multiple children are affected, which are like shaded in red. And this family has come, the mother is again pregnant for the fifth time, and this family is a consanguineous family. And they want to know the recurrence risk for this particular recurrence risk of the same condition in this particular pregnancy. So here, first we need to understand is we need to establish a genetic diagnosis in the first four children. So we need to do a detailed evaluation of one of them, and then do genetic testing to establish a genetic diagnosis. And once we establish that, and once we establish the carrier status of the parents, then the recurrence risk in this pregnancy is also 25%. So something that we need to understand here is that the recurrence risk is 25% in every single pregnancy. So it does not mean that if somebody already has had two children affected, three children or four children affected, that they may not get another affected child. The chance of recurrence is 25% in every single pregnancy. So this is what we should be able to communicate to the family. So there could be another scenario where a consanguineous couple comes to us for pre-conceptional counseling. So as in any other situation in genetics, in a genetics clinic, we should take a detailed family history and try to understand whether there is history of any genetic condition in the family. And generally, the background risk for having a child with a congenital malformation or autosomal recessive disease or a stillbirth or any genetic condition is around 2 to 3% in 2 to 3%. This is for general population. The risk in first cousins to have an affected child with an autosomal recessive disease, stillbirth, or congenital abnormalities is only a mild increase, and it comes to around 3 to 5%. And so this we should be able to communicate properly to the family uh, about this minor increase in risk of having a child with any congenital malformation or a rare autosomal recessive disease. And we could offer this family something known as carrier screening, where for common autosomal recessive conditions. And we should also give them information about the availability of expanded carrier screening, where next generation sequencing technique would be used to identify pathogenic or directly pathogenic variants in any rare autosomal recessive condition. Now, this is another situation where where a child in a particular family. So here you can see a family where there is second degree consanguinity. and here the uncle has apparently married his niece, and they had a child who had neuroregression and ichthyosis. So here, when genetic evaluation was done, this child, it turned out that this child had two different autosomal recessive conditions, and one was GM2 gangliosidosis, and the other one was ichthyosis. So both these two different autosomal recessive conditions were apparently diagnosed in this particular child. Now the family wanted to know the risk of having an unaffected child. Like they did not, they wanted to know their chance of having a child who neither had neuroregression nor had ichthyosis. So a Punnett square, the one that is shown on the right side, will help us in identifying, in understanding the recurrence risk in such a particular condition. So here, this particular family has a 56% chance of having an unaffected child, having a child without neuroregression and without ichthyosis. So this is in contrast to family where only one autosomal recessive condition is there, where I already said that 75% of the time they would not have any phenotype. So here, with two autosomal recessive conditions, the chance of them having an child with neither of these disease is around 56%. So it is important actually to understand the number of autosomal recessive conditions and then modify our counseling accordingly. Thank you.